Hey everybody, Professor Long here, doing your A&P lectures. As you guys know, the coronavirus or COVID-19 has colleges and campuses across the state of Texas shut down. So we're having to move to an online setting. Some of the lectures that I need to give require a much larger format, so I'm filming in a classroom during the limited time that I have available to me. Most of the video lectures have been done in shorter lectures at home on a smaller board, but thank goodness I have the opportunity to use the equipment I need to use for today. So please bear with me. I'm filming this in two ways. I'm using my laptop and my cell phone to try to get this into YouTube. The laptop, the lighting doesn't look quite as good, so I'm going to do the two videos, check them out, upload the best one. Whichever one you're seeing was the better of the two. So please bear with me. We're going to do the best we can to get through all of this. So now, in my Part 1 A&P, Human Anatomy and Physiology 1, we have been covering muscles. I've given a short series of lectures over the structure of muscle. Let me get some markers and get some of my supplies ready. Okay, so now, what we have talked about previously is this. Okay, so a skeletal muscle itself is a bundle of smaller structures called fascicles, and this muscle is covered by an outer connective tissue covering called the epimysium. Inside of that, there are some smaller round structures called fascicles. And technically, we could say that a muscle is a bundle of fascicles. If I pull one of the fascicles out, each fascicle is covered by a connective tissue covering called the perimysium. We've covered this before as well. And that perimysium surrounds some smaller structures, which we're going to call muscle cells or myofibers. So technically, a fascicle is a bundle of muscle cells or myofibers. Now, when we look at this, if I pull one of these myofibers out or a muscle cell, what we see is along its length is the outer covering, which would be the cell membrane. Because it has unique properties, the cell membrane of a muscle cell is referred to as the sarcolemma. Now, each individual skeletal muscle cell is surrounded by an outer connective tissue covering called the endomysium, and it individually wraps each individual skeletal muscle cell. The endomysium of every muscle cell is tied together with all the others within a fascicle at the end of the fascicle. The perimysium, which is made of the same dense connective tissue as the endomysium, is going to be tied in or woven in with all of those, and each fascicle is all woven together at the end of the muscle. And since that's the same connective tissue that we see covering the muscle, then that forms the tendon, and these lines would go out and literally kind of braid it all together. So this is the tendon at the end of the muscle. Now, one of the things that we talked about in a previous video is that a muscle cell is a bundle of smaller, rounder subunits called myofibrils. And so there's a bunch of myofibrils, hundreds of them inside of a muscle cell. I'm going to pull one out and make it a little bit thicker so that we can look into it, okay? And each myofibril does not have a connective tissue covering. I've drawn it sort of as a solid straight line, but as you'll see from looking at some of the models in lab, that it's really sort of a bunch of proteins that are interlocking or sticking together. And it's almost as if I had a wall here and I stuck a bunch of spears in it and they were sticking out towards me, almost like my arms sticking out like this, okay? And then if I had another wall and I had some spears sticking out this way, they would start to overlap each other. And then on the other side of this wall, I had more of the same spears, and I just keep repeating the subunits. Those are what are going to make up what we call a sarcomere. And as you learned if you watched the previous videos, the sarcomere has a specific structure and layout, and they're very highly organized. We see some Z lines here. This would be a Z line, and this would be a Z line. And then on the other side of each Z line, there will be other of these smaller proteins called the thin filament or actin sticking out, and these units repeat throughout the length of the muscle. In the middle of the sarcomere is the M line. And as you would have learned in a previous video, the M line has proteins sticking in both directions, which is called the thick myofilament or the thick filament, and sometimes used interchangeably with the word myosin. And this sort of arrangement of Z line, M line, Z line, M line, Z line, M line, so on and so forth, with actin and myosin overlapping, is the repeating subunits. Now, if I look at the end of a myofibril, if I cut it in an area that we called the zone of overlap in the previous video, I would see some actin and myosin filaments kind of overlapping each other and sticking in here. Now, one of the things, I'm going to draw this quickly and erase it, but if I could cut right at a zone of overlap, there's a very unique and mathematical, almost mathematic arrangement. It's been studied by mathematicians, where every red dot here represents a thin filament or actin filament, and they are all kind of strung around like this. Right in the center of 6 of these is one myosin or thick filament. So one myosin is surrounded by 6 actins. And I can almost connect them like this, although they would only be connected to each other at the Z lines. Now, the myosin filaments, 3 of them surround one actin. And at an M line, they would all be stuck together somewhat like this. And if I increase the number of these, oops, I didn't draw that very well to begin with, you can see this 6 around 1 or 3 around 1 arrangement. Every myosin is surrounded by 6 actins or thin filaments. Every actin is surrounded by 3 myosins. Now I could extend this out, and it'll take a second, but just bear with me. You can see how I can continue this arrangement and now this myofibril continues to get bigger and bigger and bigger the more actin and myosin I add. I can add another 3 myosins here. I'm not going to do this ad infinitum, but here's a myosin, here's a myosin, here's a myosin. 3 myosins surrounding an actin, and you can see how large one myofibril can get. But these proteins are arranged kind of overlapping each other in a very 3-dimensional structure. This is as if I was looking at it on end. Here we see it looking at it from the side. So this view is like looking at my arm or my fist from this angle, and then this view is looking at it this way. And these repeating subunits continue, sarcomere after sarcomere after sarcomere, throughout the length of the myofibril. Now, the goal of this lecture is for us to look at the molecular structure of actin and myosin and try to understand how action potentials work so that we can understand how how action potentials influence muscle contractions, how neurons cause skeletal muscle fibers to contract and control them with neurotransmitters. So, one of the things that we did not add in the previous lecture is this. And you'll see this when you look at the lab videos, that every myofibril is surrounded by a membranous structure, an interconnected network of membranous structures. In typical cells, we call it the endoplasmic reticulum. In skeletal muscle cells, because it has a somewhat unique feature in that it has an extra number of calcium channels and it has the ability to conduct some electricity, we call it the sarcoplasmic reticulum. Just like the cell membrane of a muscle cell is called the sarcolemma instead of the plasmalemma, well, the endoplasmic reticulum of a skeletal muscle cell is called the sarcoplasmic reticulum. The same is true in cardiac muscle. Now, in our models, they use this yellow color to indicate the endoplasmic reticulum. And it is literally draped around here, wrapped around all of this, and all these interconnected tubules. I don't know if you can see this orange marker very well or not, but there's a bunch of interconnected tubules literally wrapped around the myofibril, as if I took a sheet of paper and I wrapped it around my arm. This is going to be interrupted by a tubule that extends down and through the muscle cell so that things could pass through the muscle cell, called a T-tubule or transverse tubule. And so I'll add a transverse tubule here and a transverse tubule here. So sections of sarcoplasmic reticulum are interrupted by transverse tubules, and then I would have another section on this side going this way. Okay? So there'll be sarcoplasmic reticulum, T-tubule, sarcoplasmic reticulum, T-tubule, sarcoplasmic reticulum, T-tubule, sarcoplasmic. And the sarcoplasmic reticulum is very uniquely arranged over these sarcomeres. Now, we're going to come back to this drawing, so I'm going to leave it up. But what I want to look at is the molecular structure of actin and myosin for a moment. So imagine this. Let's say I cut a small section of this myofilament right here, the thin filament, actin. I cut a small section out and I magnify it. Now, what we find out is that these large molecules of actin, called F-actin, F stands for filamentous. Proteins, when we synthesize them from transcription and translation at the ribosome, are just long chains of amino acids. I'm going to erase this really quickly, but if I have a whole bunch of amino acids in a long chain, some of these amino acids have side groups that might cause two of them to want to bind together. And so the amino acid structure, the protein, can fold up and form chemical bonds. And they'll fold up into specific three-dimensional structures. If when they're folded they look like long thin lines or filaments, they're called filamentous proteins. If they ball up in little globules, they're called globular proteins. Well, when scientists were first studying actin, they called it actin, or the thin filament, but it turns out that it's really a bunch of smaller proteins that are strung together. So if I were to look at the molecular structure of actin, it's made up of smaller subunits. 2, 4, 6, 7. And it's actually 2 rows of these subunits stuck together. And they're stuck to another protein that sticks right down the middle. We're not going to talk about connectin and titin and get into all of that level of detail for now. I'm trying to get the basics, the fundamentals down. But this is a long chain of small subunits that makes up a piece of F-actin. Each individual subunit, so this whole piece here, is called F-actin. Each individual subunit is called a G-actin molecule. So one F-actin molecule is made up of a number of G-actin molecules. And if you see me looking at the camera funny, I'm trying to see how well you can see the colors on the board. This is the first time I'm doing a video in this room. Anyway, so these G-actin subunits each have an area on them where some activity takes place. It's kind of a binding site, and we call it an active site or a binding site. And there's a region on each one of these where some activity can take place. That area on each one of these is called the active site. The active site is where another molecule, the large or the thick myofilament, myosin, can bind. So the active site is the location on the G-actin where we call myosin heads will bind. And that's during contraction, when a muscle contracts. Okay? So, I know that's right on the edge of the screen, my apologies. So, when our muscle is going to contract, the myosin heads will grab onto these actin filaments and pull. And they'll pull the Z line towards the M line. On the other side, these F-actin filaments look the same. So if this is the M line, my body, my arms would represent the myosin molecules. The myosin molecules are going to reach up, grab onto actins on either side, and pull. And when they do, that will slide the Z lines closer towards the M line. When this sarcomere shortens, then that's going to pull on the 2 ends. When all the sarcomeres shorten, then the entire myofibril shortens. Because the myofibril is tied to the end of the muscle cell, and that's tied to the end of the fascicle, which is tied to the end of the muscle at the tendon, that's what's going to pull on your tendons to move your bones. Now, we're not done with the structures of the actin filaments. The thin myofilament, the myosin heads, which we're going to study in a second, have such a huge or high affinity. And affinity is a sort of a likeness to bind to in biochemistry. They have such a high affinity to bind to the actin subunits to the active site that, if exposed and the myosin head is sitting here, it's going to grab and bind almost instantaneously. It's just the electrochemical properties of the proteins here. So if that happened, your muscles would stay contracted and cramped 24/7. We would be literally paralyzed in a giant cramp, like when you get a Charlie horse, it's not usable. So in order to prevent the muscle from contracting all the time so that we can control muscle contraction, there's another protein that sits over the active sites. Now, one of the things I want to show you is I've drawn these in a straight line. If you look at the pictures in the book, the molecular structure is such that these actin subunits are somewhat twisted around each other, kind of coiled in a helical structure, but I've drawn them kind of flat here. Nonetheless, there's an additional protein that sits over the active site on the actin subunits and blocks it so that the myosin heads cannot bind. That protein happens to be called tropomyosin. And one of the things that we can note about tropomyosin is that it blocks the active sites on actin. When a muscle is relaxed, I'm just going to put relaxed in parentheses here, but while the muscle is relaxed, tropomyosin is actually blocking the active sites so that the myosin heads cannot grab and pull. The tropomyosin is not directly attached to the active site itself. It is attached to the actin molecule by another protein, and this protein actually has 3 subunits or 3 binding sites, and this protein sits right on top of all of this. And I'll draw the 3 subunits sitting like this, attached to the actin and to the tropomyosin. This protein happens to be called troponin or troponin. I learned it as troponin. Some books call it troponin. It's tomato, Caribbean, doesn't matter to me. So, either way, troponin has 3 binding sites, and I'm going to list the binding sites in order of affinity. The highest affinity first, the second affinity, and the lowest affinity. So troponin can bind, or troponin can bind to 3 things. 1, it can bind to actin. It holds the tropomyosin bound to the actin. It almost never wants to let go of the actin ever. Number 2, it can bind to calcium ions. Ca2+, as you should know, is the abbreviation for calcium ions. I'm going to step out of the screen here. And so you can see that it has the second highest affinity for calcium ions. And then, you know what? I said this wrong. It has the highest affinity for tropomyosin. It has the second highest affinity for calcium, and then for actin. Sorry about that. So, essentially, the troponin molecule will bind to 2 things at once. It'll bind to tropomyosin and calcium, or it'll bind to tropomyosin and actin. If no calcium is available, meaning free floating calcium inside the cell, then the troponin molecule will hold tropomyosin over the active sites and prevent access to the active site from the myosin heads. If calcium becomes available, if I sprinkle some calcium on here, the calcium ions will bind to the troponin. As you guys should know, proteins have a 3-dimensional shape. Just like if I asked you to hold on to, say, a couple of footballs, and if I asked you to hold these footballs and you're going to get an A on this test if you hold them for the entire class period. If I started to throw things at you, I don't know, like a volleyball or a softball or a tennis ball, I threw a book at you, you may never let go because you want the A on the exam. You have a very high affinity for the footballs. If I threw you a live baby, hopefully you don't want an A so badly you're willing to sacrifice another human so that you would let go of the footballs and catch the baby. When you unbind from one thing, you stop binding to it and bind onto another, just like a protein, you change your shape and space. So what happens when calcium binds to troponin is it's going to change shape and pull tropomyosin off the active site. I'm not going to draw this on both sides. It would happen at both sides simultaneously, and these 2 tropomyosin molecules would be pulled into this groove between between the actin filaments. But essentially, if I were to sprinkle some calcium here, what would happen is this part of the molecule would change its conformation. It would pull the tropomyosin off to the side this way, and I might see the troponin over here bound to calcium ions sticking out. So anytime calcium binds to troponin, troponin changes shape and pulls tropomyosin off the active site, exposing the active sites. Okay? So I'm going to leave this like this because I don't want to keep redrawing it. Now, we need to look at the structure of myosin, and then I'm going to stop the video so that I can keep them to a manageable length, and I'll do an additional video that's going to go through the actual muscle contraction, how we control it with neurons. So the structure of myosin is a little bit simpler than actin. The actin subunits have their active sites, which are covered by tropomyosin, and troponin is holding tropomyosin over the active sites while the muscles relax. If we sprinkle calcium, calcium binds to troponin, it will somewhat release from the actin and slide over and expose the active sites. The myosin molecule is much simpler. So if I cut a section of this myosin molecule out here out, what I'm going to see is they kind of resemble little golf clubs. Maybe not so much a real golf club, but there's a part of the myosin molecule that does this. These two parts right here are called the heads. The heads can also be called cross bridges, and it has two of them. Scientists have worked out the molecular structure of these proteins, and so what we're going to see is that the head of the myosin molecule has the ability to bind to the actin. This part of the molecule is called the hinge. The hinge has the ability to bend, and it will allow the myosin molecule to reach up and bind the heads or the cross bridges to the active site on actin. And then it's going to allow it for what they call the power stroke. It's going to flex and pull. Since the Z line would be over here, it's going to pull the Z line towards the M line at this end. And then this entire portion here is referred to as the tail of the myosin filament. So the myosin molecules, the individual molecules, have two cross bridges or heads which bind to the active site, the hinge, which allows for flexing and the power stroke, and the tails. And it turns out that if I look at them, a number of these molecules are sitting like this. So I have a whole bunch of them stacked together so that if I were to look at this structure here, I would see all these little myosin heads sticking up and sticking in either direction. As a matter of fact, they stick off in three directions because one myosin, actually it's six directions, because one myosin can grab onto six actins when we looked at that molecular structure earlier. So now, all that I have to do is expose the active site on actin,